Alright guys, welcome back to Underground Science, and in this video we're going to be talking about the Lac Operon, okay? So this is in E. coli, okay? The Lac Operon is present in E. coli bacteria, okay? And it's actually a pretty cool mechanism. First, let's get familiar with the genes, okay? So this gene right here in green, right? This section of the genetic material is the promoter. This is where the RNA polymerase will bind, okay? And then we have the operator that's right in front of it. Okay, then we have these three genes that will code for, or that will end up basically after you, or if you express these three genes, it will end up being proteins. Okay, like if you undergo transcription and translation, we're going to have proteins from these genes. So this yellowish gene, after you undergo transcription and translation, will have beta-galactosidase. If you express this LAC-Y gene, right, after we undergo transcription and translation, we'll have permease, and LAC-A gene, right, after it undergoes translation, we'll have trans-acetylase. For now, let's just focus on beta-galactosidase and permease, okay? Now, but beta-galactosidase is cool because what it's going to do is it's going to break down lactose, okay? Beta-galactosidase will break down lactose into glucose and galactose, okay, because that's what lactose is made up of. It's a disaccharide. Permease is going to actually make an opening in the bacteria so that you can have lactose go into it, okay? So it's going to make a opening in the bacteria, and this opening will cause lactose to go into the bacteria, okay? So that the bacteria can use the lactose for energy. All right, so again, permease, right? Beta-galactoside permease is going to basically create an opening into the bacteria cell wall, right? It's going to attach in the bacteria cell wall, create an opening in the bacteria so that the lactose outside of the bacteria can go from outside to inside of the bacteria so the bacteria can use the lactose for energy, okay? And then we won't focus on trans-acetylase for now, okay? Because the main things I think we should know is LAC-Z and LAC-Y and what proteins they end up coding for, okay? So we went over the promoter, operator, LAC-Z, LAC-Y, and we know that this is LAC-A, okay? Now behind the promoter, we have the cap site. And behind that, way behind it, we have the LAC-I gene, okay? This is going to code for a repressor protein, okay? So what's going to happen is we're going to have the LAC-I gene will be making a repressor protein, okay? So let's say we represent this repressor protein as a triangle. This repressor protein, what it's going to do is it's going to bind onto the operator, okay? So if it binds onto the operator, and we said what? RNA polymerase will bind onto the promoter, right? And we know RNA polymerase will undergo transcription, okay? Or well, is the enzyme that's gonna basically take all the DNA and convert it to RNA, right? So if RNA polymerase, if we represent that with just like right here, right? Because we know green is for the RNA polymerase. If RNA polymerase binds here and it starts to go, right? It can't because the operator has the repressor on it. So RNA polymerase is blocked. It cannot go and perform transcription. Therefore, you can't express these genes, which means you cannot make these enzymes. Okay, so now we understood, okay, if there's a repressor present, then that means it's bound to the operator because that's where the repressor will bind, which means RNA polymerase cannot advance itself and start expressing, right, these genes, start transcribing these genes. All right, so that's that. And so we have to go over a few scenarios to make sense of this. All right, so let's go and look at the first scenario. The first scenario is, let's say we don't have lactose. What will the bacteria do, right? Will it turn on this Lac Operon or will it not, right? Will this Lac Operon be expressed or will it not be expressed, I should say, right? So think about this, right? If we don't have lactose, right, why would you want to make enzymes involved in breaking down lactose? Because we said LAC-Z, for example, codes for beta-galactosidase, and we said that breaks down lactose. Well, why would you want to code for beta-galactosidase, or why would you want to make beta-galactosidase if you don't have any lactose to even break down? So if you don't have lactose, right, just by knowing, right, just by making sense of it, you can say like, okay, well, the Lac Operon will be off, right? We're not going to have any expression of it, okay? But if we actually look at like in detail why, right? If lactose is present, what happens is it gets converted into an isomer of lactose called allolactose, okay? So when lactose is present, what happens is lactose can get converted into an isomer of lactose, which is called allolactose. Okay, so let's say we represent this allolactose, right, as a green circle. Okay, allolactose will go and bind onto the repressor that's present on the operator, right? And what that's gonna lead to is the repressor will leave the operator. Okay, it's not gonna be bound to the operator anymore. So let's represent that like this, right? So then we're gonna have the allolactose and repressor right here, and the repressor is now, or the allolactose is in green, okay, and the repressor is in red, right? So once allolactose binds to the repressor, there's nothing on the operator now, right, that's blocking the RNA polymerase from transcribing these genes. So that means RNA polymerase is free to go. That's only if there's lactose present, because if lactose is present, that means allolactose is present. If allolactose is present, then that will bind onto this red triangular repressor, right? If allolactose binds onto the repressor detaches from the operator, and now there's nothing on the operator blocking the RNA polymerase from transcribing these genes. Okay, so you'll make these genes. You'll make enzymes required to break down lactose. Okay, so that's why, but if we don't have any lactose present, that means we don't have allolactose to bind to the repressor, okay? That means the repressor will stay bound to the operator, which means the RNA polymerase will continue to get blocked, okay? Because it can't go because the repressor is bound onto this operator, so the RNA polymerase cannot go forward and transcribe these genes. Therefore, you're not going to make these genes. You're not going to make the necessary enzymes to break down lactose. Which again makes sense, because why would you want enzymes to break down lactose if you have no lactose, right? Okay, so no lactose, that means you're not going to have expression of the Lac Operon, so it's going to remain off. Okay, let's look at our next scenario. Lactose plus a bunch of sugar, bunch of glucose. Okay, so remember we said lactose is what? Lactose is glucose and galactose. Okay, it's a disaccharide. Okay. Glucose is just glucose, right? So if the bacteria had an option to use lactose as energy or glucose as energy, it's going to choose glucose, right? Because you don't have to break down glucose. It's already ready to use as energy. But lactose, it'll have to break this bond between glucose and galactose, right? So that why would it spend energy doing that if it already has glucose for energy use, right? So if you have lactose, but you also have a bunch of glucose, let's see how that's going to affect this, right? So we said if you have lactose, you're going to have allolactose, which means what? If you have lactose, you'll have allolactose, which we said allolactose is in green right here. The allolactose will bind to the repressor, therefore freeing the repressor from the operator, and RNA polymerase will then transcribe the genes, right? So Lac Operon will be on if we have lactose. But on top of that, we're saying we also have a bunch of glucose. Okay, so let's see how that affects us. All right, so one thing we should mention, right, is that glucose, right, a lot of glucose will actually inhibit adenylate cyclase. Okay, a lot of glucose will inhibit adenylate cyclase. Now, how is that important? Because remember, adenylate cyclase did what? Right? It converted ATP into cyclic AMP. Okay, so that means if you have a lot of glucose, you're going to have very little cyclic AMP. Okay? And what happens is if you have very little cyclic AMP, you have very little of cap proteins. Okay, remember we had that cap site? Well, this is where it comes into play. All right, so again, if you have a lot of glucose, you'll have very low cap proteins. All right, so why is that bad? Or not necessarily bad, but why, like what effect does that have, right? So again, if you have a lot of glucose present, then the bacteria won't make many cap proteins, okay? You'll have very low number of cap proteins. And why is that important? Because remember, we said what? This site was called the cap site, right? This site right here was called the cap site. Cap proteins bind to the cap site. So this cap protein, right? Let's say this is a cap protein, okay? This cap protein binds to the cap site, and this helps RNA polymerase to transcribe these genes. So again, if cap proteins bind to the cap site, this helps RNA polymerase transcribe these genes, okay? So the Lac operon is expressed when cap proteins bind the cap site. But we said if you have a lot of glucose, right, that's going to lead to a low number of cyclic AMP, which will lead to very little amounts of cap proteins, which means not many cap proteins will bind to the cap site, which means RNA polymerase won't really transcribe these genes, okay? So that means if there's a lot of glucose, we won't express the Lac operon. Why? Remember, the Lac operon is present so that we can make enzymes to break down lactose. But remember, we said that's easier just to use glucose as energy because lactose, right, we have to break a bond in order to get two monomers, right? So if we have a lot of glucose, why would we want to use lactose, right? So let's just not express the Lac operon and use the glucose first. So that's why if you have a bunch of glucose, right, we're gonna try to turn off the Lac operon through this cyclic AMP and cap protein mechanism that we just discussed, all right? But the thing is, right, so what do we say here, right? We have a lot of glucose, okay? That means we're going to have very little cyclic AMP, means very little cap proteins. If you have very little cap proteins, you won't have cap proteins binding to the cap site. That means that RNA polymerase will not express these genes, right? It will not transcribe these genes, which means Lac operon will be off. But we also said in this scenario, what if we have lactose also? So the high glucose situation is trying to turn off the Lac operon, but the lactose situation, remember, if we have lactose, allolactose will bind to this repressor, which will free the repressor from the operator, which means RNA polymerase can then express these genes. So if we have a lactose, that's trying to turn on the Lac operon, and a bunch of glucose is trying to turn it off. So that means we're going to have very, very low expression of the Lac operon, okay? So we're going to have very low expression of the Lac operon, okay? Because both are antagonistic to each other, right? High glucose is trying to turn off the Lac operon, and lactose is trying to turn on the Lac operon. So we're going to basically end up basically not producing many of the enzymes, right? We're going to end up expressing Lac operon like not much at all, okay? So it's going to be a very low expression of the Lac operon. But what if we had lactose but barely any glucose, okay? So we said that high glucose will lead to low cyclic AMP. So that means low glucose will lead to a bunch of cyclic AMP, which means we're going to have a bunch of cap proteins, right? So low glucose means a lot of cap proteins. A lot of cap proteins means we're going to have a bunch of cap proteins available to bind to the cap site. The more the cap proteins bind to the cap site, the more this Lac operon is turned on. So the more cap proteins bind to the cap site, that means RNA polymerase will want to transcribe these genes even more strongly. So that means low glucose, right, turns on the Lac operon very heavily. And we already know lactose turns it on heavily too. So if we have two things that are trying to stimulate the crap out of the Lac operon and turn it on, that means we're going to have a very increased amount of Lac operon expression, right? So Lac operon will be heavily turned on in this scenario, all right? So that's basically it, guys. I think these three scenarios should put a good idea into our heads about the Lac operon, right? And yeah, that's the Lac operon. I hope you guys learned something in this video, and hope to see you guys in my future videos, all right? Thanks for watching.